by Dr. Brett McCray from the University of Michigan, and it's going to be a talk about biomarkers in CMT, the clinical outcome assessments, and how they can be used for clinical trials.
All right, thanks, Charles, and thanks for the opportunity to speak today. So I'll just jump right in. I still try. There we go. So we just heard a great talk about a successful clinical trial in ALS, and I'm actually going to start this discussion with an unfortunately failed clinical trial in CMT that many of you will probably remember from about 10 years ago. So this was a trial of ascorbic acid or vitamin C for CMT1A. And so this was a really disappointing result because not only did the trial not show a treatment benefit, but it also showed that the chosen outcome assessment here, the CMT neuropathy score, really failed to show any expected disease progression. So if you look at this graph on the right, you can see that the placebo patients actually showed improvement on this scale, which was not what was expected compared to the dotted line, which is what they expected to see. So this really showed that this was not a good outcome measure and really highlighted the challenge of trying to detect progression in a slowly progressive disease. And so this really prompted a big shift in how we thought about clinical trial readiness in CMT and really prompted several natural history studies to try to understand better what we could measure and what changes in CMT, and also a lot of work to try to identify and validate and improve different clinical outcome assessments and identify new disease biomarkers. So we heard a little bit about kind of the pathways for drug approval, but I kind of wanted to really highlight sort of in a simplistic way what I think the FDA sort of thinks about with drug approval. So generally what they're trying to find is drugs that improve clinical outcomes. And so these are supposed to measure how a patient feels, functions, or survives. And so we have clinical outcome assessments that measure how a patient functions, and we have patient-reported outcome measures that measure how a patient feels. So when we're thinking about CMT and the challenges of kind of using these metrics, we know that CMT is a really generally slowly progressive disease, so that presents a challenge. And then clinical trials are typically 6 months, 1 year, 2 years, so a relatively short period of time, and we don't see a lot of overt progression in CMT. And so our COAs and PROs that we would use in a trial really need to be responsive over this time frame. So then there's what we just heard about, surrogate endpoints. And so these can be used in place of clinical outcomes in certain contexts when the outcome is going to take a long time to develop and you have a validated surrogate endpoint that can predict that outcome. And so unless you consider neurofilament as that potential surrogate, we don't really have those in CMT. But what is available is the FDA Accelerated Approval Program, where you can use as we just heard about, a non-clinical outcome, a surrogate biomarker that's reasonably likely to predict a clinical outcome. And then eventually you need to show that biomarker or that surrogate endpoint does actually predict that clinical outcome. And so longer studies are used to then show that there is clinical benefit. So what makes a good outcome measure in CMT? And so for the COAs and the PROs, we really need to be thinking about measuring how a patient feels or functions, because fortunately in CMT, most forms do not affect lifespan. For biomarkers, these need to be reasonably likely to predict some kind of clinical outcome. And in either of these cases, what we want is a really responsive assessment. And so what this means is that you get a large change in the surrogate endpoint or outcome assessment over a certain period of time. And the way we typically try to measure this is through the standard response mean, which is essentially the change in that outcome over a certain period of time divided by the standard deviation. So essentially a signal to noise over a period of time. And the convention in thinking about SRM is that less than 0.5 is a low responsive measure. A moderate responsive measure is 0.5 to 0.8, and greater than 0.8 is strongly responsive. So what we would like are COAs and PROs that are surrogate endpoints that are in this high SRM range for CMT. So the COAs that we have in CMT I wanted to go through now. So one of the older ones is the Overall Neuropathy Limitation Scale, or ONLS. And so this is kind of a blunt instrument. It's a 12-point scale with higher scores meaning more disability. It's somewhat simple, 0 to 5 for arms, 0 to 7 for legs. And so this was validated in 100 patients, but only 9 of those had CMT. So this really is not a CMT-specific clinical outcome assessment. In the original study, they did find a pretty good SRM, about 0.76, so just shy of the strong responsive range. So this was used in a clinical trial, the FARNEX trial, for CMT1A, and to my knowledge, these results are not fully released, but we know that it didn't meet its clinical endpoints, and at least from what has been shared with the public, the company seemed to suggest that perhaps part of the reason the endpoint wasn't met was because the ONLS didn't really perform well and may have been subject to some subjectivity. So the CMT neuropathy score is another COA that we've used quite a bit for natural history studies and somewhat in trials. And so this is a score that includes patient report of sensory and motor symptoms, as well as physician sensory and motor scores, and then electrophysiology. Each of these subparts is scored on 0 to 4. 4 represents higher disability. And this was really designed with CMT in mind, so it's very heavily weighted toward distal sensory loss and distal upper and lower extremity dysfunction. So if you take all of the components, you have the CMT neuropathy score, but that includes electrophysiology, which we don't really like to repeat frequently. And so a lot of times we end up using the CMT-ES, which includes all the components except for electrophysiology. And then you can also look at the CMT symptom score, which is just patient-reported symptoms. And so after the failure of the ascorbic acid trial that I mentioned that used the CMT-NS, sort of went back to the drawing board and tried to improve the CMT NS and created a version 2. And then a subsequent study in CMT1A used Roche analysis to further try to improve the CMT-ES and CMT-NS. And so these have now been evaluated in multiple natural history studies. And so if you look at all comers for CMT1A and the new CMT-ESR, the Roche weighted, the performance is still quite poor actually in a 2-year study. The SRM is only 0.17. However, if you look at patients stratified by baseline severity, you can see that the CMT-ESR actually perform the best in patients with mild disease at baseline entry into the study. And so if you look at mild or moderate patients, you can see that the SRM does improve somewhat and it actually reaches the moderate responsive range for the mildly affected patients. So these scores were also used in natural history studies for CMT-X1, where again, the performance was low over a 1-year period for all patients, but improved when you looked at mild patients specifically over 1 year, and then out to 3 years, it was strongly responsive. Similar findings were in CMT2A. The CMT-ES performed a little bit better as this patient population tends to be a little bit more severely affected. And then finally, in CMT4C, through a natural history study, again, kind of low responsiveness over 2 years when you looked at all patients. So still some problems in responsiveness for the CMT-ES, no matter what sort of iteration you're looking at. So this kind of prompted a development of new outcome assessments. And so initially, there was the generation of the CMT pediatric scale, which was designed obviously for children, and this was then adapted to make an adult version, and that's the CMT functional outcome measure, or CMT-FOM, which we've heard about a few different times. And so this includes a range of different assessments of strength, function, ambulation, and balance. So it's, each item is scored 0 to 4, with 4 being more severe disability. And then after the raw score is generated, you get a scaled score from 0 to 100, again with higher scores indicating more severe disability. And so these are just some of the components. You have strength tests with a dynamometer of hand grip, ankle dorsiflexion and plantar flexion strength. Upper limb function is assessed with a couple of tests, one to flip a peg over and another to put small pegs into a pegboard. Lower limb function has a few different assessments. So there's sitting to stand, climbing stairs. Initially, there was a 10-meter walk run, but this was actually eliminated because there was some misfitting and some inconsistencies in scoring. There's multiple measures of balance, and then mobility is tested with 6-minute walk test and a timed up and go test. So as far as PROs for CMT, the only adult PRO that's specifically designed for CMT is the CMT Health Index. And so this was really based on querying patients about what symptoms they felt were most important in their lives. And the result was 105 questions across 18 different themes. And for each question, patients are asked to rate how severely this particular problem impacts their daily life. So this was used in a short trial of follistatin for CMT1A, and CMT-X. And at least in this short study, in this kind of small study, there was not a clear change in the CMT-HI over time, so it didn't seem like there was clear progression in terms of patient-reported symptoms. However, as I think we'll probably hear more about in the next talk, in a trial for SORD neuropathy, the CMT-HI does seem to be showing some signs that it is detecting a meaningful benefit with the therapy. So perhaps this is going to be a useful marker going forward. So as far as the SRM and performance of the FOM and the HI, we are waiting for results from the Accelerate Clinical Trials in CMT study, or the ACT-CMT study, which we've heard some of those preliminary results, but the final results are not released yet. But once we get those, we'll be able to get a sense of how the FOM and the HI are performing longitudinally. And so this is a study looking at CMT1A in patients followed over 3 years. It also looks at some other of the outcome assessments I mentioned, as well as some biomarkers that I'm going to tell you about. We do have some data on the CMT-PEDS, which is the pediatric version of the FOM. And again, looking at all patients, you see moderate responsiveness at 2 years. And as similar to CMT-ES, if you stratify patients based on mild baseline severity, you see improved performance of this outcome assessment. So on to biomarkers. So at this point, I would say muscle MRI fat fraction has really been the most promising, the most reproducible, the most responsive measure that we have in CMT. And so this is based on a protocol that measures the amount of fat in muscle, with the idea being that as you have axonal degeneration, you get denervation of the muscle, and the muscle gets replaced by fat, and that reflects kind of disease progression and severity. And so this has now been validated as a measure in CMT1A. It's almost exclusively been used in the lower extremity, but it's been looked at in the foot, the calf, and the thigh. At the calf, in a cohort of CMT1A patients, over 1 year, there was a significant increase in fat fraction, and the SRM performed very well with a SRM of 1.04. And then if you stratify patients, eliminate those that have really low baseline fat fraction, the SRM performance of this metric is even better. So this has now been looked at in all of the most common forms of CMT, and so this is the data I just showed you for CMT1A. MRI fat fraction has also been evaluated in CMT-X1 with a 1-year SRM in the moderate range. CMT1B performing a little better and almost to the strong responsive range, and also in CMT2A. And if you aggregate all these patients, and now this is data over 1 year showing really strong responsiveness compared to the outcome assessments that I mentioned, most of which only showed moderate responsiveness at 2 or 3 years. So another biomarker that's particularly important for CMT1A is looking at PMP22 levels in tissue. And so it turns out many labs have tried to do this, and it's technically quite challenging. But John Severin's group was able to show that if you take skin tissue from the forearm and normalize to other Schwann cell genes, you can get really robust and reproducible results that show the expected elevation of PMP22 levels. And so this could be really useful as a marker of target engagement, and maybe even a way to titrate a therapy to get the optimal knockdown of PMP22. So we just heard a lot about neurofilament light, and this has been studied quite a bit in CMT. And so in general, there have been in different papers pretty consistent mild elevations in neurofilament if you kind of aggregate patients versus controls. What you'll notice is that depending on the study, there is certainly some degree of overlap between patients and controls. So for a given patient, they may or may not have elevations of neurofilament. There's also been efforts to see if the elevations predict clinical severity or progression, and those have also been somewhat mixed. So it seems that neurofilament levels don't consistently show elevations that give you any meaningful data about severity or progression. So other serum biomarkers have been evaluated and identified, most of these in CMT1A. And so one is TMPRSS5. And so this is actually found as a very specific biomarker for CMT1A. It was not elevated in other forms of CMT. So it's a Schwann cell protein that has been shown to be elevated in patients in multiple cohorts and seems to perform better in terms of sensitivity and specificity versus neurofilament light. There's also been several microRNAs, again, that were looked at in CMT1A specifically. And so there are a number of microRNAs, some of which are Schwann cell specific and some of which are muscle specific, which may reflect denervation, that seem to be able to distinguish patients from controls. And many of these perform similarly in terms of sensitivity and specificity compared to the other biomarkers that I've mentioned. NCAM1 and GDF15 were biomarkers that were found actually in screening patients with different forms of CMT and also animal models of different forms of CMT, including type 1, type 2, and X. And so these are kind of thought to be somewhat subtype agnostic biomarkers. And so these were found to be elevated across multiple different types of CMT. And unlike many of the other biomarkers that have been validated, they actually seem to show some ability to discriminate patients based on severity, with higher levels seen in patients with more severe disease based on the CMT exam score. So in conclusion, with this initial failed trial in CMT1A, there's been a development of a lot of different COAs. There still remains the challenge that CMT is slowly progressive, and even the most perfect clinical outcome assessment may not really be able to detect much change because there may not be much clinical change over a 1 to 2 period of time. We do know that kind of uniformly in these various studies, when you stratify patients on baseline severity, looking at more mild patients, it seems that these outcome assessments perform better. And so it seems that patients who are mild at inclusion have a greater rate of change by these assessments. That comes with the potential trade-off of limiting recruitment if you're going to use that data to power your trials just with mild patients. In terms of the FOM and the HI, we should have more data soon about how they perform longitudinally, but at least they've been used somewhat in clinical trials and seem to be performing reasonably well. Of the biomarkers we have, MRI fat fraction seems to be the most promising, the most responsive. And particularly patients that have a moderate baseline fat fraction seem to show the greatest 1-year change. One of the key questions is, what does a change in muscle fat fraction have to do with how a patient feels or functions? And that remains to be determined. And certainly how that would be viewed by the regulatory agencies is also unclear at this point. And then we have a number of biomarkers that are promising, that can distinguish patients from controls, but we don't know how those would respond to an effective therapy. We do know that in some cases these have been tested in animal models where effective therapies do lead to reductions in NFL and NCAM1, I think we heard about earlier, but we don't know how that's going to correlate with clinical trials. And so it's going to be really important for us to continue to have discussions with regulatory agencies about how these different biomarkers could be used in trials and whether they could be used to really support a drug approval. So with that, I can take questions if we have time.
We have time for, I think, one question, if there is one. Over there, at the other side.
Thinking about instruments or biomarkers across different forms of CMT as opposed to really thinking about them within specific forms of CMT, given how heterogeneous it is.
You're asking to develop more specific biomarkers, or disease-specific, or less disease-specific?
More specific to the genetic form of CMT, because I guess my working assumption anyway is that the instruments that work best in one are not necessarily going to be the best in another, and that's maybe even more the case for biomarkers.
Yeah.
So should we really not be so much thinking about what are good biomarkers overall, but what are good ones for specific forms of CMT?
I think that's a great point. I mean, I think 2 great examples of that are SWORD, obviously that as we'll hear about, that clinical program was really accelerated because of the specific biomarker. HSAN1 is another one where toxic sphingolipids are produced and can be released into the bloodstream. And so I think there's opportunities in certain types of CMT where you can have really specific biomarkers, and those are probably preferable to these more general biomarkers, but not every form is amenable to that level of specificity.